Cell Animation Project by Tariq Abu. We will now move into the phases of the cell cycle. The first phase would be the G1 phase, where cells increase in size, produce RNA, and synthesize protein. It is also an important phase because a control mechanism is activated. It also ensures everything is ready for DNA synthesis. The next phase we will move into is the S phase. The S phase is to produce two similar daughter cells. To complete this, DNA instructions in the cell must be duplicated. DNA replication occurs during the S phase. We will then move into the G2 phase. During the G2 phase, the gap between DNA synthesis and mitosis is closed. The cell will continue to grow and produce new proteins. At the end of this gap is another control checkpoint to determine that the cell cannot proceed to enter mitosis. The first one being prophase. Chromatin in the nucleus begins to condense and becomes visible in the light microscope as chromosomes. The nucleus disappears, centrioles begin moving to opposite ends of the cell, and fibers extend from the centromeres. Some fibers across the cell form into meiotic spindle. The next phase of this cycle would be metaphase. Metaphase is when spindle fibers align the chromosomes along the middle of the cell nucleus. The line is referred as the metaphase plate. This organization helps to ensure that the next phase, when chromosomes are separated, each new nucleus will receive one copy of each chromosome. We will then move into anaphase. The paired chromosomes separate and move to the opposite sides of the cell. Motion results from the combination of the kinetorch movement along the spindle microtubules and through the physical interaction of polar microtubules. We then move into telophase. Chromatins arrive at opposite poles of the cell and new membranes form around the daughter nuclei. The chromosomes disperse and are no longer visible under the light microscope. The spindle fibers disperse and cytokinesis or the partitioning of the cell may also begin during this stage. Showing examples of the telophase. And the last phase would be cytokinesis. In animal cells, cytokinesis results when a fiber ring composed of a protein called actin around the center of the cell contracts, pinching the cell into two daughter cells, each with one nucleus. In plant cells, the rigid wall requires that a cell plate can be synthesized between the two daughter cells. Now we move into cancer cells, and we look at how cancer cells can affect the cell cycle. Cancer can happen anywhere in the cell cycle. It just depends on what goes wrong and causes cancer. It can be things like smoking, radiation exposure, and it affects the cell cycle by the rate of cell division. Genes are there are two genes that can affect the cell cycle, these being proto-oncogenes and tumor suppressor genes. Proto-oncogenes are a group of genes that cause normal cells to become cancerous when they are mutated. Tumor suppressor genes play a critical role in regulating when cells are allowed to divide and increase in number. When DNA damage is detected in a cell, some tumor suppressor genes can stop the cell from multiplying until the damage is repaired. In cancer, mutations can cause an overabundance of growth signals or a lack of reliance on reserve signals, so the cell can divide in an unregulated manner. Thank you for watching my presentation.